Hello everyone. In this series of videos, I will explain the process of photosynthesis. This is the second video in the series and I am going to explain the regulation of stomatal opening and closing. Okay, so first we need to understand what are stomata. They are tiny microscopic openings present on the surface of plant tissues that facilitate gas exchange and regulate water loss. Okay, here gas means oxygen and carbon dioxide. Okay, so we are going to learn few things about stomata. So stoma, this word comes from the Greek language and it means mouth. Okay, stoma is the singular form and stomata is plural. Fine. So first we need to understand how does a stoma looks like. Fine. So as I have told you that stoma are nothing but a small opening, right? They are encircled by two kidney shaped cells named guard cells. Okay, guard cells. And there are some adjacent cells also called epithelial cells. Fine, they are epithelial cells. These guard cells have chloroplast. Okay, it's mean they can perform photosynthesis, right? They can perform photosynthesis. So whenever the guard cells become turgid or swollen, the stomatal pore opens. Stomata opens. Fine. And when there is water loss from the guard cell, they become flaccid and stomata is what? Closed. Right now. Now, in your syllabus, there are two theories regarding stomatal opening and closing. Fine. First one is sugar concentration theory. First one is sugar concentration theory. This one is comparatively easier. So, in this theory, what happens? In presence of sunlight, I have already told you that stomatas are what? Photosynthetic cells. It's mean they can perform photosynthesis. Whenever in presence of sunlight they perform photosynthesis, accumulation of glucose, that is a sugar, right, occurs. It's mean now these cells are dense in what? Glucose. Now these cells are dense in glucose. Fine. And hence they become hypertonic. Okay. Hence they become hypertonic. And we all know if these two cells, these two kidney shaped, remember guard cells are kidney shaped. Okay. Now when these two kidney shaped cells become hypertonic, what will happen? Water from adjacent cells will enter here. water from adjacent cells will enter here. As a result, what will happen? These two guard cells will become turgid, right? And it will result in stomatal opening. Okay, this was the first theory, that is the sugar concentration theory. Next, we are going to discuss about the potassium ion exchange theory. Okay, our second theory is potassium ion exchange theory. Fine. Now this one is little bit more complicated than the first one. Fine. So in this one, what happens? Let me take some another color. Okay. What happens? During daytime, in presence of sunlight, starch is converted into malic acid, right? Starch is converted into malic acid. Now malic acid is a weak acid. Fine. So we are going to follow some steps here. Okay. Now malic acid being a weak acid, it will dissociate itself into malate ion and hydrogen ion. Okay. Now here, when the stomata are about to open, the guard cells activates the proton pump. Guard cell does what? Activates the proton pump. Fine. Once the proton pumps are activated, H plus ions, okay, so H plus ions will actively pumped out, okay, actively pumped out from where? From the guard cells, okay, pumped out from the guard cell. Now it will result in a negative electrical charge, okay. If so many ions are ions are pumped out, then obviously it will become negative electrical charged, negatively charged. Okay, so to maintain the balance of ions, what will happen? To maintain the balance of ions, K plus, that is potassium ion, will rush into the guard cell. Okay, now this process is called ion exchange. Okay, ion, this is called ion exchange. It means hydrogen ions are exiting and potassium ions are entering, right? H plus ions are pumped out from the guard cells and potassium ions along with some Cl ions, they entered the guard cells. Okay, now in the next step, what will happen? In the next step, these potassium ions will react with malate. Okay, malate that was produced by dissociation of malic acid. Okay, now they will form potassium potassium malate. Okay. Now these potassium malate does what? They reduce the water potential. Fine. It means the cells become what? Hypertonic. And we all know when guard cells are hypertonic, then what will happen? Water from the epithelial cells or the adjacent cells will enter the guard cells, right? And the guard cells will become turgid again, like this. Lots of hydrogen ions are pumped out. Potassium ions are entering right now, and potassium ions, they are doing what? They are reacting with malate and making potassium malate. As a result, it is reducing the water potential and cells are becoming hypertonic. To maintain the balance, water from adjacent cells will enter the guard cells and the guard cells will become turgid again. And whenever the guard cells become turgid, it's mean what? Stomatal pore is open. Now these two were in your syllabus, that is sugar concentration theory and potassium ion exchange theory. Okay. In our next video, I am going to discuss about the mechanism of photosynthesis. Mean what are the reactions taking place in light dependent phase and light independent phase. Okay, thank you. That's all for today.